So Dr. Faith Walker is here with us today. She is a research professor at Northern Arizona University, and she is the director of the Species from Feces Lab. Love that lab name. She has worked with non-invasive genetic samples and eDNA for 25 years. Her team employs a variety of tools in genetics and ecology to understand population biology and natural and evolutionary history of organisms. So Dr. Walker will be sharing her presentation on detection of listed bat species under bridges through the use of fecal DNA. So thanks, Dr. Walker.
All right, thank you, Andrea, for that introduction. And thanks everybody for being here to another talk about poop right before lunch. So today I want to give you an introduction to the Species from Feces program and talk a little bit about its history, and then describe a project that we have about bridges in the US and Canada and identifying bats, and then describe a new method about detecting bats at bridges. But first, let me introduce you to my team. This is us, both past and present. These are students and staff, and they're very excited about poop. These are the folks that work with your samples when you send them to us. And so I joined this bat team about 15 years ago, and at the time we were doing a lot of bat capture and telemetry and acoustics, but we noticed that we would go into a cave or a mine or other kind of roost, and we would often not see bats, but we would see a lot of this, guano, and guano is a wonderful thing. It doesn't fly around in the dark, and it's loaded with DNA. So my task was to develop a genetic assay that works at detecting species globally. And so we did that by targeting a region of the mitochondria called CO1. It's about 200 base pairs long, and it's called a DNA mini barcode. And it's really good at species level discrimination. So it's kind of like a supermarket barcode, except for it detects bats. So of all the bat species in the world that have genetic data available, for 92% of them, we can get to the species level with this DNA mini barcode. And this is what it looks like on a global map. So the blue are those bats that we can get to the species level. The kind of orange is those we can get to the genus level. So that means that there are bat species within a genus that share a DNA mini barcode and can't be distinguished between with this marker. And then the gray are species that haven't been barcoded yet. So lots of species in say Africa, Asia, Australia need genetic work. And the cool thing about this system is that it's scalable. So you can take a single guano pellet and determine who pooped it, like Jason and Annie did, or you can take pooled guano, so say 200 fecal pellets from around a roost, and determine all the bat species that contributed to it. You can also, while you're at it, throw in other markers to look for, say, microbes or arthropods, so you can get at disease or diet while you're at it. And so we came out with two papers. The first was Species from Feces, which described the assay in 2016. And then a few years later, we published a fecal sequel, which tested the limits of the assay for bat species ID. Sorry, I have a thing about names. All right, so you can go to our website and type in nau.edu/batDNA, and you can find our search tool where you can put in your species of interest and be determined and be returned the capability of our assay. So here, for instance, I typed in Myotis odalis, and it says, yes, we can determine this species with this barcode. And so we use this DNA mini barcode for our own projects in mines and caves and other roosts. And then we got some interest from other folks wanting us to screen their samples. And so we started this fee-for-service program that we called Species from Feces. And so here's an example of a project. So these were mines in the Four Corner states. And this figure here shows you 41 mines. So each bar is a mine at which we collected a single pooled guano sample. And then the y-axis is the proportion of reads or the proportion of sequences that belong to each of these bat species that are color-coded. So we detected between 1 and 4 bat species per mine, which was a lot better than what we could see visually going into those mines. We then started working with bridges. And so what I want to show you today is a project that we're collaborating with Wildlife Conservation Society Canada. So we have bridges in the US as well as Canada. There's 500 that we collected guano from 2021 to 25. And Cory Olson is analyzing this. He's going to be pushing out a paper. And so it's not completely analyzed yet, but I can give you a glimpse of what we have. So the objectives were to examine how bats, and in particular listed bats, are using bridges and whether they're important for conservation and management, to determine the characteristics of bridge roosts, to find day roosting bats and especially maternity colonies, and to use guano sampling at bridges for genetic analysis. So to collect baseline data to monitor changes in bat distributions, and then also to get insight into bat diets and collect baseline data to examine changes in prey populations over time. And then also to monitor for PD, which is the fungus that causes white nose syndrome, by testing the guano. All right, so in Canada, there were all kinds of different types of bridges, and the data, again, haven't been analyzed yet, but this is what we have. They particularly like these adjacent box girders, and those were where the maternity colonies were found. And then also they like the I or T-beam bridges with open soffits. And for the other bridge types where there was mixed materials, they really liked the concrete sections as opposed to, say, metal or wood. All right, so here is a bridge with expansion joints. This is where the bats hang out, like some of these other talks have shown. And in this case, there's a maternity colony in one of these expansion joints. If you peer into that on the right here, you can see what they look like they're looking out. And so that's what we're calling a day roost. And then as Riley pointed out, they also use bridges at night. So they're coming back to roost and poop. And we actually got more poop from these vertical surfaces than we got from the ground. But we did collect it off the ground below these expansion joints and also on the vertical surfaces. And to do that, we use these telescoping poles on which we have fixed these little buckets, and we use those to grab the guano. All right, so this is the western provinces of Canada, and the percentages are the percent of bridges that had guano. And they're color-coded by the amount of guano present. So the darker colors are a very high amount, so say 5 cups or more. And then the lighter pink are negligible, so between 1 and 10 fecal pellets per bridge. And here are results from 2023. So I flopped these figures on the side. So now bridge is on the y-axis and the proportion of reads is on the x. And you can see a lot of green. So the green in this case is Myotis lucifugus. It's considered endangered in Canada, and it's listed by the IUCN. And then our other listed species was Myotis septentrionalis, which was found in the Yukon. And here are results from 2024. This time, excuse me, gray represents Myotis lucifugus, and green represents Myotis septentrionalis. So we found septentrionalis in the Northwest Territories. And here's a map showing the US as well. So we have, okay, so I basically collapsed all the non-listed bat species into the color green. So those green bits of the pies are not listed. And then the other colors are listed by some entity or another. So we have a lot of Myotis lucifugus across the landscape. And then in the Midwest, you have that blue piece of pie, which was Myotis totalis. There's also a bit of tricolored bat in there. And then on the east, we have, we have the gray bat. All right, so let's talk about diet. So again, these data aren't analyzed yet, but this is what we have. We detected 379 genera, 433 species. And if you rank them by frequency of occurrence, the top two were Lepidoptera and Diptera. So not too surprising, but we'll get more into that later. And I just wanted to have an aside here. So my team has pivoted our work with bats to anything that poops. We do a lot of ungulate diet work. So just so you know, in case for other projects, if you need our help, we're happy to work with other species as well. And then the last thing I wanted to talk to you about is airborne eDNA. So we were involved in this study in 2022, which used active air filters. So these are air filters that are attached to a battery, which runs a fan that pumps air through a filter. And Nina Garrett and Beth Claire, they brought these air filters to Belize, put them out in the rainforest, and then gave us a piece of the filter, and we ran DNA metabarcoding to see what we would get. And we found some bats, birds, a gecko, and a bunch of domestic animals. So we were pretty excited about it. This is one of the first studies that showed that we can use this out in the open to detect biodiversity. But we were wondering, well, what would happen if we just used a passive air filter to make it easy on our field team? So here we just used food grade cheesecloth suspended by PVC piping, and we brought it to Nicaragua. We had open air sites where we netted bats just to figure out what bats are in the area. And then nearby, we had our stations with the passive air filters. We also had an enclosed site, which was a cave roost. And here's what we found for open air. So on the right-hand side, these are the number of individuals netted of all these species. And then on the left is the number of detections with airborne eDNA. So we had 4 detections. So it worked, but it wasn't great. And I think the take home here is if you're going to be looking to detect bats with air in the open, you should be using an active air filter to get as much air as you possibly can through that filter. But the story was different in the cave. So on the right-hand side were the number of species that we visually saw, and there were 2. And then with the air filters, we were able to detect 5. So I think in this situation, when you're in a cave, it's fine to use a passive system. And a couple other people have found this as well. Just when their active filters failed and they extracted the DNA, and they were finding just as many bat species in those as the active ones. Okay, so what's next? Well, we're taking these primarily the active air filters and suspending them from bridges and determining how guano differs from airborne eDNA, differs from acoustics in bat species detection. And I want to thank these people and entities. And here's our QR code if you want to learn more about the Species from Feces program. Thanks. Any questions?
Great, thanks, Dr. Walker. Yes.
Hi, I'm just curious if that online search, does that include the like diet organisms or just the bats?
Just the bats.
And then if I could follow up, do you keep all the data that's like sent to you? Do you guys end up kind of owning it? Like, is there kind of that clause when people send you in samples?
There is, right. We don't have that clause and we don't own it. So like when Cory publishes, I'm not going to include many of the US sites.
Okay.
Yeah.
And I didn't see any beetles in the diet, right?
Oh, I don't know. I'd need to look at it again.
Okay, just.
Yeah, I'd be surprised if there weren't beetles, but
Yeah.
I could be wrong.
Thank you.
Thank you for the talk. On your map, it looked like there was not a lot of examples to the Central Plains into Texas. Is that just not receiving them, or was your barcode not able to detect anything?
That's just not receiving samples from those areas.
Is there any want or push or help that you could employ those people to do that, or something else?
Well, we'd be happy to analyze from those areas. Yeah, we could talk about it if you're from, are you from? Yeah, okay. Yeah, we could talk.
There, so since you brought it up, there are a number of data gaps, particularly for bats, across the Central Plains. And so any opportunity to fill in those information gaps is really critical. So yeah, please reach out to folks across these sessions who you think could potentially help you all.
Hi, Faith, great talk. I was curious, I noticed Cory bat popped up in your, in some of the bridge stuff. Do you have, your collaborators have any ideas about why Cory bat might have popped up, since they're not typically considered a bridge roosting species? But then I have heard some sort of anecdotes about them popping up at bridges. I'm just curious if you guys thought maybe there was a contamination, not contamination, but like it flew in from somewhere, or if you felt pretty confident that the samples were like actually dropping from the bridge.
Yeah, I think we're pretty confident. There were a lot of reads for Hories, so, but that, I don't know. That's all I know. Riley, did you find Hories?
No, I didn't. But there's evidence, a couple papers in Yellowstone that have Tories.
Yeah, I've seen one paper with Cory's like night roosting at a bridge, but
Yeah.
yeah.
I think some of it could be like a temporary route of weather, and all things that they're like, help, I need to find some place to go, so, yeah.
Yeah, we see them, especially during migration. Cories and silver hairs will stop at bridges and culverts and just be seen once and that's it.
I feel like that needs to end up in the literature.
Yeah, sure.
I bet they feel tight.
On buildings as well.
Yeah.
And the same, it's migrating through and just be like, oh, yeah, you just stay here and then take off, something like that.
What is the shelf life of the samples? Or if we were seeking funding and we want to start collecting samples during regular field work and store them, is there a period of time in which they degrade? Or
So if you store them in a freezer, they'll be good for years and years and years. Yeah.
Great questions, everyone. Thank you again, Dr. Walker.
Thank you.